Hi, today we are here at the Plant Biology Society meeting at the poster session. Here is Dr. Kirti Rathore from Texas A&M University. He has been working with carton biotechnology and he has some excellent research and we have some special guests actually here with us, and here there are African delegates who came all the way from African countries, different countries, and they came here to part of their workshop and follow-up meet with the international committee that was organized by Leon Koshian. Here is Marianne Quain from Ghana, and here we have Lucky Omaigui from Nigeria, and we have here Richard Akinwale from Nigeria, and we have Remy, and you're from Nigeria?
Yes.
Yes. Okay, and then we have here David from Nigeria, and then we have Moses from Nigeria. A lot of Nigerians. Well, the main goal for the African delegates here is to interact with the U.S. Scientists, and in today now they're going to introduce the work that Dr. Rathore is doing, and then talk about their work, and hopefully they can end up with some potential collaborations for future. And we also actually have some of Dr. Rathore students. I'll let Dr. Rathore take over from here and tell us, introduce what you are doing at Texas A&M and in your lab.
Okay, so by the way, this is Srinath and Dave. They are working in my lab. And let me just kind of talk about the work. So this research pertains to cotton seed improvement. And you may be wondering why improve cotton seed farmers grow cotton for fiber, not for the seed. But the fact is that the plant produces more seed than it produces fiber, and it also is very rich in protein and oil. So about 23% protein and about 21% oil, and and we cannot utilize that source of protein very efficiently right now because cotton seed contains gossypol, which is toxic to people like us, and also monogastric animals like chicken or fish. You cannot feed that cotton seed to these animals. And so, but at the same time, this gossypol protects the plants from insects. And the gossypol is present in these dark colored glands throughout the cotton plant, in the leaf, in the stem, in the flower, and also in the seed. Now, all these other places, it serves a very useful function in terms of protecting the plant from insects and diseases. So what we have done is to utilize the tools of molecular biology to shut down the pathway for gossypol biosynthesis, but only do so in the seed, not in other parts of the plant. Okay, and so that's why, and you cannot do these kind of things through traditional breeding. So that's the advantage of using these molecular tools, is that we can do this kind of thing in a very specific way, at a specific time, in a specific cell, a specific tissue. And so we've been able to shut down this gossypol biosynthetic pathway and have brought down the level of gossypol from something like, it's about 1% in the seed. From 1%, we brought it down to about 99%. I mean, we reduced it by 99%. Yeah, so it's very, very low gossypol. Okay. Now, this is a kind of a follow-up work which we have done. We already published our original work in 2006, and this paper came out, and what this paper deals with is that how solid is the technology. You know, everything works beautifully in the greenhouse, but what about when it, when you face the real life challenges? Is it still solid? And so this paper was just about to see whether how solid this technology is. And it's a technology we have used, is gene silencing technology called RNAi. And so we wanted to see if it'll be stable. That was one thing. From generation to generation, is it stable? And so this chart here shows that stability of the trait. So we had, these are the gossypol, these are different lines, and these are the gossypol levels in the first generation. Okay. And then second generation, they come down to very low level, and then they stay stable. In different lines, you can see initially in the T1 generation, which we call the first generation T1, they will be a little bit high because it's going to be a mixture of seeds. It's not going to be all the seeds which have our transgene. It's going to be a mixture of seeds. But in the T2, then it should come down, and it has. You can see that they stay down. So we've tested these things for 5 generations in the greenhouse, and everything is looking good. The trait is stable. Now, following this work, which we have not published yet, and we are going to publish that soon, we have also studied 2 lines out of these 9 lines. We have taken 2 lines and we've studied those in the field for now 3 generations, 3 years, and again the trait is stable and everything. So that was the number 1 point. We wanted to make sure that because this is brand new technology, we did not know whether it will work from generation to generation. So now we've gone up to 8 generations and the trait is stable. So we're pretty confident that this will work well under field conditions. Okay. Now the other thing that we wanted to see was again, most of it is not here because this is a follow-up study. We wanted to see what about leaf and flowers and all these other parts, petiole and so on, brag tissue, which the insects like to chew. How much gossypol do they have? And that study we have done, and again it shows that all these other parts of the plant contain normal levels of gossypol and terpenoids. So, and even under field conditions, we've tested all these things, and it's working okay. So what that shows is the trait that we have introduced into cotton is very specific. So the silencing is very specific in the seed only. It does not happen in other parts of the plant. The other thing that we wanted to see was how are these seedlings going to respond when they're challenged with, say, disease. So when the young seedling is growing in the field, obviously there are all kinds of diseases and everything. How are the seedlings going to respond? Now, cotton has this property that it, when it's challenged with some pathogen, its gossypol levels and the levels of other terpenoids that are derived from the same biosynthetic pathway, they go up. And these are supposed to protect the plant from diseases and so on. So that's what we have studied here, and it's kind of very complicated kind of result here, but this black bar here is the non-transgenic plant, and the 2 other bars here are the transgenic ones. And when we challenge these plants with Rhizoctonia solani, you can see that there is an induction, a little bit less, but there is still an induction. Because and again, there are different kind of gossypol related compounds that we have studied, and again you can see that in the root these are induced. So what that means is that even a very young seedling has not lost its ability to launch this defense response when it sees a pathogen attacking it. Okay, so that's what that is about. And we have done, this is a little more kind of just evidence for the RNAi silencing working. And we also looked at the gene level to see, so this is at the biochemical level. We also looked at the gene level, whether the gene itself that we have shut down in the seed, would it be activated when you challenge the plant with a pathogen? Okay, and that's what this diagram is. So so number 1 here is the non-transgenic, and 2 and 3 are the transgenic lines. And when this is under normal conditions, no infection. Okay. But when you infect the plant with Rhizoctonia solani, you can see this induction. So this is in the non-transgenic and in the transgenic. So we can see induction as early as 2 hours following the infection. That means that the normal mechanism of the plant to launch this defense response is still functional. Okay, so this particular poster is just about that. So our first paper, which came out in 2006, just showed us that this technology can work. Now you may be wondering, what is the benefit of this thing? Okay. Now, I think again, most people don't realize it, and I did not realize it until I started working with cotton, that worldwide uh, the annual crop of cotton, we produce 44 million metric tons of cotton seed. Okay, that amount of cotton seed has about 10 million metric tons of protein. Now, if you could utilize that protein for, say, directly for human consumption, you could meet the basic protein requirements of 500 million people. Half a billion people can be nourished by cottonseed if it was possible to eat it directly. And now I know that most people probably would not eat cottonseed. But you can take the same cottonseed and say feed it to the chicken or feed it to fish. A lot of aquaculture is going around now, so people are using fish grown in aquaculture to eat. And so our hope is that this cottonseed, we can take that and now can start feeding it to the chicken or the fish. And now you may be wondering why not feed it to the cows. Now cows can handle a little bit of gossypol. Okay, so cows are not hurt as badly as say people like us or chicken or fish and so, but the important thing to remember is that the cows are not very efficient in terms of converting feed into meat or milk. It takes 6 pounds of feed to get 1 pound of beef, whereas when it comes to chicken, it's about 2. You give it 2 pounds of feed, you get 1 pound of chicken meat. And some of the fish is even more efficient. You give it 1 pound of feed, you get 1 pound of fish meat. Okay, so if you have limited source of protein, vegetable protein, it's better to take that protein and feed it to the chicken or fish than to feed it to the cows. And so that's where we see the benefit of this technology. So again, this thing has come from very basic science, but we've taken an extra step and try to utilize those basic science tools to do something useful. And the other thing to remember about this is that farmers will continue to grow cotton for the fiber, not for the seed, but at the same time you have this massive resource which right now is not being used very efficiently, but now that cottonseed becomes useful for all these other animals, and someday maybe even for human beings. In fact, in the 50s, the plant breeders came up with a gossypol-free cotton plant. Okay, and at that time, a lot of studies were done, including in the U.S., even in my university, and in South American countries and Africa, where they took the cottonseed from that particular plant, which did not have gossypol, and they found that it's a good source of nutrition for human beings. They've done studies with children, women. There was no problem. It could be a good source. Now, the reason why that failed, even though it was a good product, the reason why that failed was that there was no gossypol in entire plant. So the insects were attacking those plants, and so the farmers did not want to grow it, and so for that reason it failed. So our technology is a little more refined, that we have only done this gossypol elimination in the seed. Other parts of the plant remain the same.
So that was a great work, and a great introduction. Actually, this is just the beginning of I guess the potential benefits that we can explore using biotechnology, and it's a good example here with this low gossypol cottonseed. And I hope all of our African delegates have learned something new in biotechnology projects going on in U.S. And hope you can follow up with Dr. Rathore here and see if you can fit in your objectives of your institutes, respective institutes, and Dr. Rathore's program at Texas A&M University. And with that, thank you all for being here today.